In this lecture, we're going to discuss control of microbial growth. So first, let's talk about some terminology. And the definitions may be a little bit different than what you are familiar with using. So sterilization means removing all microbial life. Everything is gone, completely clean. So we can do this in the lab, we do this in medicine, we can do this commercially. The goal of sterilization is to kill all of the microbes and their endospores, because remember, endospores are more hardy and more resistant to killing. And so, especially with food, Clostridium botulinum is a bacteria that makes botulism toxin, which is really prevalent in food poisoning. So it's important to prevent that bacteria from getting into food. Okay. Disinfection means removing harmful microbes. The key here is from a surface. So if you're disinfecting something, you're disinfecting a non-living surface. This usually does not eliminate endospores. It does not make things sterile. You can use chemicals, UV radiation, boiling water, or steam for disinfection. Antisepsis is removing pathogens from living tissue. So sepsis means microbial contamination, and asepsis is the absence of that. So antisepsis would be removing microbial contamination. Sanitization means lowering microbial counts, so reducing the number of microbes on a surface. Not eliminating them by any means, but reducing them to a low enough level where they won't hurt you. For example, restaurants, when they sanitize their tables or their glassware and their dishes, they're lowering microbial counts, but they're not sterile, but the microbes aren't at a level where they'll harm you. Biocide and germicide, anything that ends in side is going to be something that kills something, right? Biocide kills living things, germicide kills germs, bacteria and viruses probably. There's viricide, fungicide, anything of that manner. And then we have bacteriostasis, which we already talked about a little bit when we talked about growth of microbes, but bacteriostasis means you're inhibiting but not killing the microbes. And by inhibiting, we mean that they're not growing, they're not metabolizing, they're not making waste products or toxins or anything like that. They're just kind of stopped where they are, right? Because static means stop. Once that bacteriostatic element, whether it's cold or a chemical, once that's removed, bacteria can grow again because they weren't killed in the first place. When we talk about microbial death, we can talk about rate of death. For the most part, bacteria, when they are heat treated or killed with an antimicrobial or chemicals, they die at a fairly constant rate. And even if that rate is 99%, you can go from a number of 1 million down to just one living cell in as little as 6 minutes. So that is a pretty rapid decrease. Okay, we're not going to worry about the math of all of this, but essentially if you decrease by the same percentage each time, you will get down to a very low number fairly quickly. And because that happens quickly, we can plot it on a logarithmic curve. Again, we're not going to get into log scales and graphs and that kind of thing in this course. All right, so let's talk about how effective different treatments are and what can change that effectiveness. So the number of microbes that you start with is a big one. It's a numbers game. If you start with a small population, it's going to be faster to kill. If you have a higher population, it's going to take longer, right? That's pretty intuitive. Environmental factors, things like temperature, pH, different disinfectants will work better at higher temperatures or lower or higher pHs. Whether that bacteria is in a biofilm or not. We know that when bacteria are in a biofilm, they're much harder to kill because of that protection that the biofilm offers them. And another thing, particularly with treating infections in living tissue, is the organic matter that's present, or even on a surface. If there's also a lot of organic matter on that surface, that can get in the way and kind of stop the chemicals from reaching the microbes effectively. The time of exposure is also a big one. If you look at the back of a can of disinfectant wipes, whether it's Clorox wipes at home or the special disinfectant wipes that you use in the clinic or other areas, they always say to wet the surface completely and to leave it wet for a certain amount of time. If the surface does not stay wet for that amount of time, then it won't be 99.9% effective or whatever it is that they're advertising their efficacy. And different chemicals take different amounts of time, but pay attention to that. I encourage you to read the labels on the disinfectants that you use on a regular basis and see what that fine print really says. How can you really use these to their most effectiveness? And of course, our last one is microbial characteristics. Every different species of microbe is different, which is why it's so complicated sometimes to try to put them under one category. Different microbes are easier or harder to kill than others. All right, so we are going to focus on the actions of microbial control agents, essentially, any of the things that we're using to try to kill bacteria. How do they kill the microbes? And we're going to focus on bacteria. So how do they kill bacteria? Well, there are a few different categories of ways that chemicals typically kill bacteria. The first one is that they make the membrane more permeable. So if things can get through the membrane easier, then they can get into, chemicals can get into cells easier. And if you can damage the plasma membrane, you can kill the cell. So alteration of membrane permeability is a big one. Damaging proteins is another big one. If you can cause the proteins to denature, they've lost their function, right? If you denature all of the proteins in a cell, that cell is dead. Or you can damage the nucleic acids. If you can damage the nucleic acids, you can stop the cells from replicating or making proteins from their genetic material. So 3 main ways: membranes, protein damage, and nucleic acid damage. All right, a little bit about the microbial characteristics and some generalizations about which ones are easier and harder to kill. So this is a really interesting graphic I found that lists some different kinds of microbes, from most resistant to killing at the top to least resistant to killing, easiest to kill at the bottom. So good news for coronavirus. Coronavirus is a virus with a lipid envelope around the outside of it. They are fairly easy to kill, but they're also small and easy to spread. With effective hand washing and disinfection and sanitization, we should be able to eliminate the spread of viruses with lipid envelopes, at least from surfaces. Okay, gram-positive bacteria are the next easiest to kill. They have that thick peptidoglycan layer, but they don't have an outer membrane that the gram-negatives do. So this thick peptidoglycan layer has teichoic acid coming up and through it, holding it all to the cell. And that teichoic acid is very easily damaged by penicillin. And that's one of the reasons penicillin is so effective at killing gram-positive bacteria. And we have other viruses, fungi. Gram-negative bacteria are way up here, above the middle of the graph. That second membrane, the outer membrane on gram-negative bacteria, makes it much harder to kill. Protozoa, cysts of protozoa are kind of similar to endospores of bacteria. We'll talk about them later. But they're very hard to kill. Endospores of bacteria are way up here. And then the most resistant and hard to kill are a thing called prions that we haven't talked about yet, but we will briefly. They are proteins, they're misfolded proteins that are essentially pathogenic. Here's an interesting genus of bacteria, the mycobacteria. So these bacteria, you can't gram stain because they have a really, really waxy mycolic acid around the outside of their cells. And they are pretty bad pathogens like Mycobacterium tuberculosis and Mycobacterium leprae. They are very difficult to kill because of that waxy layer. bacteria that make endospores are much, much harder to get rid of. Think about how easy it is for C. diff to be transferred from one patient to another in a healthcare setting. A lot of that is because C. diff can make endospores, and those endospores can survive on surfaces and in guts a lot longer than living cells can, and they can reactivate once they get into back into a healthy environment. Healthy for C. diff would be the human gut. So imagine how easy it is to possibly pick up endospores off the surface of something and get them into a body. All right, here's a little bit about prions. They're proteins that are infectious. They can be, you can get an infection by eating prion-infected meat. Prions cause essentially mad cow disease, if you've ever heard of that. So what that is it's proteins in cells in brain tissue that misfold and they clump together and they cause other proteins to clump together. And then you get this cell with all these clumps of proteins in it and it's not functional anymore and the cell dies. And this spreads throughout the tissue and causes holes in the tissue, the spongy, kind of spongy looking, like this picture in B right here. So it's referred to as bovine spongiform encephalopathy, or mad cow disease. So here's a picture of a healthy brain tissue, a brain infected with prions. See the big difference there? There's so much cell death within the tissue that it's spongy. And the biggest thing about prions is that you really can't get rid of them. There's no good drug for them. They're resistant even to autoclaving because autoclaving will, as we'll see in a minute, autoclaving will denature proteins, but these proteins are already misfolded. So it's really not going to change that much at all. All right, a couple of important things to point out here. Some disinfectants are classified by their thermal death point or their thermal death time. Thermal death point is the lowest temperature that will kill all the cells in a culture in 10 minutes. And thermal death time is the minimum amount of time it takes to kill all the cells in a culture at a given temperature. So these are related in yet different terms. Thermal death point looks at the temperature that you need. Thermal death time looks at how much time you need at a given temperature. And then there's decimal reduction time. And we bring this picture back here. Decimal reduction time essentially is how many minutes does it take to kill 90% of your population at a given temperature. So this first, every kind of interval that we show here is a 90% killing. All right, let's talk about sterilization. There are a couple of different ways to achieve sterilization. Essentially, the most effective and most common way is moist heat. We can achieve moist heat in a few different ways. By boiling is a really common one. It denatures proteins. Okay, that high heat denatures proteins. Boiling will kill most pathogens in about 10 minutes. So that's good news. However, viruses and endospores can survive boiling even for much, much longer periods of time. You actually have to get it hotter than boiling to achieve death of endospores and some viruses. So in order to get water hotter than its boiling point, you have to use an autoclave. And what an autoclave is it's, I'm gonna move on but come back to this. What an autoclave is it's kind of like this big metal chamber that produces really, really hot steam. And water normally turns to steam at 100 degrees Celsius. In an autoclave, they take this chamber and they pressurize it. It gets pressurized a lot. And so much so that the water and the steam can get really, really hot without the water completely evaporating. So instead of water turning to steam and that steam being at 100 degrees Celsius, that steam can now get up to 121 degrees Celsius, which doesn't sound like a lot, but it really is a big difference. And that amount of heat for a long enough amount of time can kill endospores. The pressure increases the temperature that steam can get up to, and then it can kill much more effectively. Okay, so autoclaves are really commonly used. We use one in our lab. They're used commonly in medicine and in dental labs and in research labs. It's kind of the gold standard for sterilization of supplies. So here's a little more detail about autoclaves. You don't need to know this though. All right, so while we're talking about sterilization and moist heat, we should also talk about pasteurization. You've probably heard of pasteurization before. Most of the milk that we consume and buy at the grocery store is pasteurized milk. What pasteurization is it is heating a substance to just hot enough for a very short amount of time to reduce the amount of microbes in that substance that would cause it to spoil. So the goal of that is to heat it for a very quick amount of time at the lowest temperature that they need to get up to, so that they're not cooking the food while they're sterilizing it. There are some organisms that can survive pasteurization, but those are unlikely to make us sick. So different products need different times and temperatures. It's usually about 30 minutes at 63 degrees Celsius. That sounds incredibly specific, but that's kind of like the gold standard for milk. So if you can get higher temperature than that without ruining your food product, you can go for a shorter amount of time. There are different kinds of pasteurization called high temperature short time, or HTST, which is only 15 seconds. And it's fairly common for packaged dairy products that can exist at room temperature. And then there's ultra high temperature at 140 degrees Celsius for 4 seconds, which essentially sterilizes things. But it's great for unrefrigerated milk and those little coffee creamers that don't need to be refrigerated. The other kind of sterilization is dry heat sterilization. So we have moist heat, which kills by denaturing proteins, and we have dry heat, which kills by oxidation, which essentially damages DNA. So we can achieve dry heat sterilization by using fire or incineration, incinerating, essentially burning things, right? Or we can use hot air. So instead of 121 degrees Celsius for 15 minutes in an autoclave on moist heat, you can use a special kind of sterilizing oven set at 170 degrees Celsius for 2 hours. You can also filter things to achieve sterilization. So if you have something that's heat sensitive, like a special media that you need your cells to grow in, and you can't expose it to heat because you will ruin all of the things in it, like proteins that are important, you can actually pass that liquid through a sterile filter into a sterile container and achieve sterilization that way. So when you make some kinds of media, usually in eukaryotic tissue culture labs, you will actually sterile filter your media rather than autoclave it. Similar process to how HEPA filters work with our air. Right, we know that low temperature can inhibit microbial growth. Refrigeration will give us bacteriostasis, but it won't give us death, right? Below freezing temperatures can make microbes dormant and will kill some of them, but not all of them, which is why we can freeze our microbes for years and then reactivate them. We can also use different things like high pressure or drying things or freeze drying things to preserve them and prevent growth of microbes for a long period of time. We can also use osmotic pressure to our advantage by adding salts or sugars to food to create that hypertonic environment. We can also use radiation. So this is really interesting. So radiation is essentially the entire spectrum of energy that exists, from radio waves all the way up to gamma rays and electron beams. And the higher you get up this chart goes backwards from right to left. The higher you go up this chart, the more ability to damage DNA these rays have, okay? So radio waves don't really damage DNA. Microwaves don't really damage DNA. Infrared, not so much. Visible light is right here in the middle of the spectrum. It's actually a very small window. And then above visible light, you have ultraviolet radiation. And we know that ultraviolet radiation can damage DNA because being in the sun too much without sunscreen can give you skin cancer, because the UV light from the sun can damage the DNA in your skin cells. Okay, and we can actually also use UV to kill bacteria by exposing surfaces to UV light. Okay, X-rays are above that, although we don't normally use X-rays to kill bacteria. Gamma rays are really often used to sterilize things, especially in industry, for like pre-packaged single-use medical supplies. So the ability of that radiation to kill microbes depends on its wavelength, its intensity, and its duration. So how long are you exposing your surface to UV light? How intense is that UV light? Things like that. There are two kinds of sterilizing radiation. One is ionizing and the other is non-ionizing. Non-ionizing is UV. Ionizing are things more powerful than UV, like gamma rays. When atoms are exposed to radioactivity, their nuclei become unstable and they can give off particles. And those particles that they can give off are essentially what are your radioactivity. And those can cause DNA damage in cells. Okay. For example, our non-ionizing or ultraviolet radiation can kill airborne microbes and disinfect medical products because it specifically causes thymine dimers in our DNA. Now, thymine dimers happen when you have two T's in a strand of DNA next to each other. It actually makes them stick together and function as one T, which it makes them stick together and function as one T, which will then completely screw up your DNA and your protein that you're trying to make from that DNA. Ionizing radiation creates hydroxyl radicals, which damage DNA and cell membranes. Right, so that's sterilization. Let's talk about disinfection. Okay, this is very relevant to our everyday lives. To achieve disinfection, for the most part, you're going to use chemicals. Okay, you're not going to get sterility for the most part. Your effectiveness, again, is going to be based on the microbes you're trying to kill, the concentration of the disinfectant that you're using, the presence of organic matter, the pH, contact with the surface, and contact time. It's called the disc diffusion method. And that is when you take a plate and you put bacteria all over the plate. And then you take these little circles of paper that are soaked in different disinfectants and you plop them on the plate and you let the bacteria grow. And the bacteria will grow wherever it can, but it won't grow in the presence of different disinfectants that are effective at killing it. Okay, so let's look at this first plate here. Around each of these white circles, there's a dark circle, right? And then the rest of the plate is kind of this milky tan color, okay? The milky tan color is where all of the bacteria has grown on the plate. The white circles are the discs of disinfectant. And these darker areas around the discs are what we call zones of inhibition. These are areas where the bacteria has not grown or has died because of the presence of whatever disinfectant or chemical are on these discs. So different discs will have different sized zones of inhibition around them, okay? For example, let's look at the middle plate. Hexachlorophene here on the left doesn't have any zone of inhibition around it. So that would be something that's not effective against this bacteria, which is E. coli. However, chlorine bleach up here has a very large zone of inhibition around it. So that would be very effective at killing E. coli. Another thing to note is that we can actually use antibiotics in the same method to see which antibiotics are effective at killing different bacteria. We have our bisphenols, things like triclosan and hexachlorophene. These are in antibacterial soaps. Triclosan used to be widely available over the counter to consumers in like antibacterial soap and things of that nature. It was actually outlawed to consumers in 2019 by the FDA. However, it is still available to healthcare and research labs and things. Another category are the biguanidines. These are things like Hibiclens, okay, and surgical scrubs. Halogens are things like iodine. They can disrupt cell membranes and impair protein synthesis. Chlorine is in this category as well. Same thing, we use chlorine for lots of things. Alcohols are really great at denaturing proteins and disrupting plasma membranes. They're not very effective on wounds, but they are effective on the surface of skin. That's why we prep skin with like an alcohol wipe. Same thing with ethanol. Ethanol we use at 70% concentration in a lot of research labs to disinfect things. You can also use heavy metals to disinfect things because heavy metals can actually cause proteins to denature. If you were to take a plate of bacteria and put old coins on it that were made of heavy metal and not just plated zinc, like a lot of coins are today, they would actually kill bacteria on a plate, kind of like those disinfectants would. But that's one of the reasons we use silver and copper in medical devices. We use silver nitrate as an antiseptic. Some medical devices are made of copper or have copper embedded in them because it helps to prevent growth of microbes. Then you have our surfactants. Surfactants essentially break up oil or grease and allow it to be washed away, which is how soaps and detergents work. For the most part, they act on a lot of different things. They have a very large spectrum. They act fast, and they're not normally super toxic, which is why washing your hands with soap and water is one of the most effective ways to prevent the spread of different germs. We have quats, like Clorox wipes, which are not made of bleach. They're made of a different disinfectant, and Lysol. They are effective against a number of different microbes by disrupting the plasma membrane. Then we have our aldehydes, like formaldehyde. We're familiar with formaldehyde because that's what's used to preserve things, right? You can also use it as a disinfectant, and it essentially kills everything it touches. We can also use it to clean different medical pieces of medical equipment. Peroxides are great disinfectants. Interestingly enough, hydrogen peroxide, they're recommending that you don't use that on wounds anymore because it can actually hurt the tissue more than it can help by killing microbes present. But peroxide is also used in some food packaging and in contact lens disinfectant. Some forms of dental hygiene use hydrogen peroxide. So there's lots of good uses for hydrogen peroxide. Peracetic acid, benzoyl peroxide is commonly found in topical acne medication because it can help kill the bacteria that cause the acne on the skin. And peroxides act by oxidizing, as you learned by looking at reactive oxygen species in our lecture on microbial growth. Okay, and then we have lots of chemical food preservatives that exist, right? They're safe in foods. I'm using air quotes, but you can't see them. Things like sulfur dioxide, sorbic acid, sodium benzenate, calcium propionate. You will see these in the ingredient label for different packaged foods. They help prevent spoilage. That's their purpose. Spoilage by mold or by growth of microbes. Sodium nitrite is a big one used in deli meats and over-the-counter, not over-the-counter, but sodium nitrate is really commonly used in the preservation of meat, like deli meat, to help prevent botulism. And then we have our antibiotics, which we're going to do next week, an entirely different lecture, used to treat diseases, obviously. There are lots of them, but they are also used in industry and in food, although much less in food than they used to be. All right, I'll leave you with this. How much disinfectant is too much? Well, as I had briefly mentioned before, in 2017, the FDA issued a new ruling banning the use of a large number of different antibacterial chemicals in consumer products because we were seeing a huge amount of antibiotic resistance that are happening as a result of overuse of these chemicals. I listed them all for you here. Triclosan is the one that I mentioned. That is the one that was commonly in like antibacterial soap and things like that. But they're all listed here, and there's a link down here if you actually want to read the publication put out by the FDA. It's like 26 pages long. You don't have to read it by any means, but it's there if you're curious. So disinfectants are good because they can prevent infection, they can prevent food spoilage, but too much disinfectant and disinfectant not used properly can lead to resistant populations of microbes that won't be killed by those disinfectants anymore. And that we'll talk about a little bit more in our next lecture. Thank you.